Welcome to SWAM NPTEL course on food microbiology for safe and sustainable food systems. Today is the third lecture in which we shall be classifying the different microorganisms. In this chapter we will be covering the classification of microorganisms, the prokaryotic cell, the cyanobacteria, then the economic importance of cyanobacteria, then bacteria, bacterial morphology, microcapsule, capsules, slime layers and sheath. The physiological characteristics are chemical changes important in food bacteriology, then cultural characteristics important in food bacteriology. So, this science of classification, especially the classification of living forms, is called taxonomy. From the Greek for orderly arrangement. The objective of taxonomy is to classify living organisms, that is, to establish the relationship between one group of organisms and another and to differentiate them. There may be as many as 100 million different living organisms, but fewer than 10% have been discovered, much less classified and identified. Taxonomy also provides a common reference for identifying organisms already classified. For example, when a bacterium suspected of causing a specific disease is isolated from a patient, characteristics of that isolate are matched to lists of characteristics of previously classified bacteria to identify the isolate. Finally, taxonomy is a basic and necessary tool for scientists, providing a universal language of communication. The discovery of 3 cell types was based on the observations that ribosomes are not the same in all cells. Ribosomes provide a method of comparing cells because ribosomes are present in all cells. Comparing the sequences of nucleotides in ribosomal RNA from different kinds of cells shows that there are 3 distinctly different cell groups, the eukaryotes and the 2 different types of prokaryotes, that is bacteria and the archaea. Organisms are classified by cell type in the 3 domain systems. In addition to differences in RNA, the 3 domains differ in membrane lipid structure, transfer RNA molecules and sensitivity to antibiotics. In this widely accepted scheme, animals, plants and fungi are kingdoms in the domain Eukarya. The domain bacteria includes all the pathogenic prokaryotes as well as many of the non-pathogenic prokaryotes found in soil and water. The photoautotrophic prokaryotes are also in this domain. The domain Archaea includes prokaryotes that do not have peptidoglycan in their cell walls. They often live in extreme environments and carry out unusual metabolic processes. Archaea include 3 major groups: the methiogenes, strict anaerobes that produce methane from carbon dioxide and hydrogen, extreme halophils which require high concentrations of salt for survival, hyperthermophils which normally grow in extremely hot environment. This is the Whittaker five kingdom classification. In this, key points are: all organisms evolved from cells that formed over 3 billion years ago. The DNA passed on from ancestors is described as conserved. The domain Eukarya includes the kingdoms of fungi, plantae and animalia as well as protists. The domains bacteria and archaea are prokaryotes. This figure shows that there are 5 categories of the kingdom. The one is Monera, which is originated from the earlier cell, that is the single cell. The earlier cell which has been converted into archaea as well as the bacteria, and this both together classified as Monera. This was further divided into the protists, in that the epicomplexans and the amoeba were further divided, and flagellates and coelates, the slime moths, the green algae, the red algae, brown algae, diatoms and dinoflagellates. These were together called protists. After that, the animals were separated, and in that it is categorized in the animalia, which contains the sponges, flatworms, nematodes, echinoderms, chordates, arthropods, annelids, mollusks. The another group which is called fungi, that is known as Mycete, which contains the molds, club fungi as mushrooms or yeasts. The another group plants that contains the seed plants like mosses, angiosperms, gymnosperms and ferns. This figure denotes that from the one that bacteria are the prokaryotes, it has divided into 3 parts, that is the bacteria which contains the mitochondria, cyanobacteria, chloroplasts, proteobacteria, gram positive bacteria, and after this is converted to archaea, which contains the extreme halophils, methiogens, hyperthermophils, and here the chloroplast is transferred here. After the archaea, the eukaryotes were generated, where the mitochondria is transferred. Mitochondria is transferred from bacteria to archaea, from archaea to this eukaryotes, and nucleoplasm grows larger, and which is converted to giardia, euglenozoa, diatoms, dinoflagellates, ciliates, green algae, plants, slime molds, amoeba, fungi, and lastly the animals. This table shows the differences among the archaea, bacteria and eukaryotes. You can see that cell types of the archaea is prokaryotic, whereas the bacteria is prokaryotic and the amoeba is a eukaryotic. The examples of this is methanosarcina, and this is the bacteria E. coli and the amoeba. The cell wall of this is that varies in composition, contains no peptidoglycan in archaea, whereas the bacteria they contains the peptidoglycan, whereas the amoeba varies in composition, contains carbohydrates. The membrane lipids, the archaea contains it is composed of branched carbon chain attached to glycerol by ether linkage, whereas in bacteria composed of straight carbon chains attached to glycerol by ester linkage, whereas in eukarya it is composed of straight carbon chains attached to glycerol by ester linkage. The first amino acid in protein synthesis in archaea is the methionine, whereas in bacteria it is formylmethionine, and eukarya it is methionine. Antibiotic sensitivity, there is no antibiotic sensitivity with the archaea, whereas the bacteria it is sensitive, and eukarya they are not sensitive to the antibiotics. rRNA loop, it is lacking in the archaea, it is present in the bacteria and lacking in the eukarya. The common arm of transfer RNA, it is lacking in archaea, whereas it is present in bacteria and eukarya. The oldest known fossils are the remains of prokaryotes that lived more than 3.5 billion years ago. The eukaryotic cells evolved more recently, about 2.5 billion years ago. According to the endosymbiotic theory, eukaryotic cells evolved from prokaryotic cells living inside one another as endosymbionts. In fact, the similarities between prokaryotic cells and eukaryotic organelles provide striking evidence for these endosymbiotic relationship. The original nucleoplasmic cell was prokaryotic. However, enfoldings in its plasma membrane may have surrounded the nuclear region to produce a true nucleus. We can see here that this is the early cell from where the bacteria is generated, and then it contains the mitochondria and the chloroplast. But this eukaryotic cells that was having the DNA, that is and it is surrounded by the nuclear membrane. This is the prokaryotic cells and eukaryotic organelles comparison. We can see that DNA of the prokaryotic cell, it has got one circular, some two circular and some linear, whereas in the eukaryotic cell it is always linear, and the eukaryotic organelles, that is the mitochondria and chloroplasts, are always circular. Histones in the prokaryotic cells, it is in the form of archaea, and it is also present and it is not present. The first amino acid in protein synthesis in the case of prokaryotic cells are formylmethionine in the bacteria and methionine in the archaea, whereas in the eukaryotic cell it is the methionine, and in eukaryotic organelles it is formylmethionine. The ribosomes, it is 70S in the prokaryotic, eukaryotic it is 80S, and in the eukaryotic organelles it is 70S. And the growth of prokaryotic cell is mainly because of the binary fission, whereas in the eukaryotic cells it is the mitosis, and in the eukaryotic organelles it is the binary fission. The prokaryotic cell. The prokaryotic cell is structurally less complex than the eukaryotic cell. They are rather small, about 1/25 of the volume of eukaryotic cell. Compartmutation, that is the compartmentalization in the prokaryotic cell, is far less than the eukaryotic cell. There are no organelle like mitochondria and chloroplast, nor is the DNA surrounded by nuclear membrane. The cells are prokaryotes with few exceptions such as mycoplasma are surrounded by cell wall. The prokaryotes are motile, moving either by swimming or gliding. The organ of locomotion is bacterial flagella. These are much simple in structure than eukaryotic flagella. The reproduction in bacteria is generally by means of binary fission. After the appropriate increase in cell dimension, septa appears starting at the circumference and proceeding inwards until eventually the two daughter cells separate. According to number of cell division and the divisions of plane, the pairs of bacteria, that is the diplococci, chains, that is the streptococci or the streptococcus, and plates, when they are divided into two planes, then it is either tetrad or the sarcina in multiple or three planes. If they divide in random planes, then they are called staphylococcus. One should keep in mind that some of these arrangements will break apart the clump together during slide preparation. Prokaryotic microbes are divided into two major groups, that is the cyanobacteria and bacteria. Now, cyanobacteria. The cyanobacteria, formerly known as blue-green algae, are prokaryotes that perform oxygen evolving photosynthesis in a manner similar to eukaryotic algae and plants. The morphology is basically that of unicells or aggregation of these to form colonies and filaments. The cell wall is indistinct. Under the microscope, they show bluish green color. Pigments present are chlorophyll A, beta carotene, phycobilin, phycocyanin, and phycoerythrin. The typical structure and different parts of the cyanobacteria is given in the adjacent figure. The vegetative growth is only known method of increase in cell number of many cyanobacteria. Cyanobacteria share many characters with algae, for example, ability to photosynthesis via chlorophyll A with liberation of oxygen. At the same time, they possess many features of bacteria like absence of nuclear membrane and membrane bound chloroplast of endoplasmic reticulum, mitochondria and Golgi apparatus, presence of peptidoglycan and diaminopimalic acid in the cell wall, infection with viruses that resemble bacteriophages, overall simplicity of bacteria. These features sharing relatedness with bacteria led many microbiologists and even phycologists to consider blue-green algae to bacteria. Cyanobacteria have captured the attention of microbiologists for two features: ability of some species to transform molecular nitrogen into a form usable by plants, thereby introducing the essential nutrients into the food chain as symbiotic partner of higher plants, for example, Anabaena with Azolla. Symbiosis means the mutually advantageous connection between people or two different organisms living together. In another word, you can say live and let live. Ability of some species to produce very potent toxin which have been responsible for death of cattle. Economic importance of cyanobacteria. It enriched the soil fertility and used as biofertilizer. The yellow pigment carotene found in algae is precursor of vitamin A. Some algae can synthesize vitamin D. Green algae contains good amount of vitamin B, C and K. Many species are used as food and feed, for example, chlorella. These are good source of single cell protein. Some algae react sharply to environment and can be used for biomonitoring of pollution. Some antibiotics are also extracted. Some algae are pathogen to man as well as to animal. Some may attack leaves of tea, coffee, pepper, mango, etc. Some may produce toxins that are lethal to fish and other animals. Some may produce neurotoxins that are lethal to birds and mammals. Now the bacteria. All prokaryotes that are not cyanobacteria are classified as bacteria. They are among the earth's most abundant and diverse organisms which are found virtually in every habitat. The cells are structurally more complex than viruses but less so than those of eukaryotes. They differ from eukaryotes principally by lacking a perforated nuclear membrane surrounding the nucleosome and hence a well-defined nucleus. A few bacteria are photosynthetic, but these differ from cyanobacteria in that bacterial photosynthesis does not release oxygen. Bacteria have influenced the life of man in various ways. Besides being pathogenic to man and animal, these are largely exploited for the welfare of mankind. Their systematic studies have fostered the growth of many independently branches of microbiology like medical microbiology, agricultural microbiology, industrial microbiology, food microbiology, molecular microbiology, biotechnology, etc. This figure shows the different parts of the bacteria in detail. That is the most obvious, that it has got the bacterial flagellum. This is the cytoplasm, it contains the plasmids. This is the nucleotide DNA which is not surrounded by the nuclear membrane. These are the ribosomes, plasma membrane is there, then cell wall, then in adverse conditions which we will see later, capsule, and this is the pilus. Bacteria include a great variety of microorganisms of different configurations and dimensions. Most bacterial species are about 1 micron and 15 micron long. Many Pseudomonas have a diameter of 0.4 micron to 0.7 micron and a length of 2.3 micron. The diameter of micrococci is only 0.5 micron. Because of their minute size, bacterial cell can be observed under light microscope with high resolving power objectives. Most bacteria are transparent and have a refractive index similar to that of aqueous fluid in which they are suspended. In order to render them easily visible, stained preparations are often used. The bacterial morphology. The shape of nearly all bacteria, with a few exceptions, can be derived from shapes like cylinders and curved cylinders. The basic forms therefore are cocci, straight rod and curved rods. Pleomorphism. Many bacteria under certain unusual conditions such as presence of antibodies, cold shocks, presence of antiserum, bacteriophage attack, etc. tend to develop an unusual form of pleomorphism, involving the formation of large spherical or distorted cells, the so-called large bodies or L form. These cells revert to their normal form if the inducing stimulus is removed after only a brief exposure. If the stimulus persists for a longer period, the cells either perish or may become stabilized in the L form. Microcapsules, capsules, slime layers and sheath. Many bacteria accumulate layers of water-rich materials on their outer surface. According to amount of material produced and the degree of its association with the cell, one of these terms may be applied, but it is difficult to define exactly borderline between them. If the material is closely adherent to the cells and detectable only with difficulty, it is known as microcapsule. If equally defined but extensive and readily visible using appropriate technique, it is called capsule. If only relatively associated with the cell, it is referred to as slime layer. Sometimes tubular structures are formed around the cells, that is described as sheath. These envelopes are usually not essential for bacterial life, but the possession of the capsules makes some pathogenic bacteria resistant to phagocytosis and enhances their virulence in animals. The chemical nature of capsules. The capsules are commonly carbohydrate, and simplest of this type is homopolysaccharide, which contains only one monomer. For example, Acetobacter xylenum has a capsule of cellulose which has only one bond type, that is beta 1,4 linked glucose. Many bacteria produce heteropolysaccharide capsule, some of which may be chemically fairly simple, containing two sugar types of glycosidic linkage, as in Streptococcus species, for example Streptococcus pneumoniae. A few species produce polypeptide and protein capsule. Bacillus anthracis produces a polymer of the unusual amino acid, that is D-glutamic acid, linked in the 3 positions. The chemical nature of slime. Much of few capsular materials may be excreted into the medium as slime. In some cases, the entire capsular material can be removed from the cell surface by shaking or homogenizing the bacterial suspension and can be removed from the medium as slime. A more extensive slime formation occurs with many microorganisms when the medium is supplemented with saccharose. A well-known example of this is Leuconostoc mesenteroides, a heterofermenting lactic acid bacterium known in sugar refining plants as a frog-spawn bacterium, which rapidly converts a solution containing cane sugar to a stiff jelly consisting of dextran. The chemical nature of sheath. Some filamentous bacteria form tubular envelopes which is described as sheath. These sheaths consist of a heteropolysaccharide containing glucose, glucuronic acid, galactose and fructose. Acetobacter aceti variety Xylinum secretes cellulose which forms the leather-like skin that surrounds the cells. The cell of Sarcina ventriculi are bound by the cellulose in regular association. The cellulose in these cases serves as a kind of mortar and is quite different from capsule, but structurally and functionally. Now, the cultural characteristics important in food bacteriology. The food microbiologists or bacteriologists are concerned with the various physical and chemical changes associated with the growth of microorganisms, which are, let us see the physical or the appearance wise changes. The bacterial growth in and on food is enough to make the food unattractive in appearance or objectionable. For example, pigmented bacteria cause discoloration on the surface of food. Films may cover the surfaces of liquids. Growth make surfaces slimy. Growth throughout the liquids may result in undesirable cloudiness or sediment. The physiological characteristics are the chemical changes caused by the bacteria, that is the hydrolysis of complex carbohydrates to simple one, hydrolysis of protein, to polypeptides, acetic acid, ammonia or amines, hydrolysis of fat to glycerol and fatty acids, oxidation reduction reactions which are utilized by bacteria to obtain energy from foods like carbohydrates, other carbon compounds, simple nitrogen and carbon compounds. Yield of products, for example organic acids, alcohols, aldehydes, ketones and gases. So, a knowledge of factors that favors or inhibit the growth and activity of bacteria is essential to understand the principles of either food preservation or spoilage. Thank you very much.